I am very happy to meet again through this video. Welcome to all. Today we are going to see the topic evolution part 2. Let us see what are the objectives today class we are going to cover. First one is geological time scale. Next, evidences for the evolution. First, let us see geological time scale. Geological time scale is nothing but it is a time scale for the origination of different life forms in our environment. Actually, generally, we divided years, month, week, days, etc. There are different years in Tamil. Okay. So likewise, in geological time scale, majorly divisible into eras. There are 4 eras are present: Precambrian, Paleozoic, Mesozoic and Cenozoic era. These eras consist of periods, epoch. The period is nothing but sub-classifications based on the millions of years. Epoch is nothing but some of the organisms are present in this area, so that is called epoch. So normally each and every species originated and dominated in the particular era. So that is called geological time scale. Say for in Precambrian period, planktons, prokaryotes are evolved in flora. In fauna, there is no animals. Then appearance of eukaryotes, then multicellular organisms, etc. So for example, in Mesozoic era, there will be the reptiles dominations. So this era is called it as golden age of reptiles. In Paleozoic era, the Devonian period is called it as age of fishes. So because here the domination of fishes. Okay. Then in Paleozoic Ordovician period, dominance of invertebrates is available. So this is called geological time scale. So one question may ask from this part, study well. So you see the diagrams. This is the Paleozoic era. So more invertebrates are developed in the aquatic forms. Okay, so you see the more invertebrates are developed in the Paleozoic era, and some fishes also developed. Ordovician period, some fishes also developed. So next, in Mesozoic era, golden age of reptiles. Okay, you see the diagram. Some reptiles are present, including dinosaurs. Jurassic period, especially the dinosaurs are dominant in Jurassic period of Mesozoic era. So next we see the evidences for the evolutions. So what are the evidences? So evidences means what? Some supporting material to describe the concept of evolutions. Previous class we defined the term evolutions. Evolution is nothing but origin of new species from the existing species, some heritable changes, some variations, that is called evolutions. What are the supportive features for the evolutions? These are called it as evidences. There are so many types of evidences. So we are going to classify the evidences in four categories. One is called paleontological evidences. Another one is called comparative anatomy. Another one is called embryological evidences. Then finally molecular evidences. So normally the paleontological evidences are called it as fossil supporting fossils. Paleontology is nothing but a study of fossils. The fossils are formed in three. One is called actual remains, another one is called petrifications, then natural molds and casts. So normally later on we see the diagrammatic representations for these evidences. Then evidences from the comparative anatomy, the structure, homologous structure, analogous structure, vestigial organs, connecting link, atavistic organs are comprising the evidences of comparative anatomy. In embryological evidences, compare the embryos, various types of embryos of organisms, start from the lower organisms to the up to the human being also. That is embryological evidences. Then molecular evidences is nothing but we provide evidences for the evolutions by means of amino acids or DNA or RNA, etc. So we are going to discuss this in next. Okay, so let us see. There are four types of evidences: fossils, and then anatomical evidences, embryological evidences, biochemical evidences. Biochemical evidences is nothing but molecular evidences. Okay, first see the fossils. The fossils, what is fossils? Fossils are the traces of or remains of dead organisms. Okay, the fossils are, so you see the diagrammatic representations. One fish going to be dead, so actually it form a fossil. Actually degradation is occurred. After the degradations it remains the fossils, actual remains. Then this is the petrifications. Petrifications in rocky substances. In the rocky substratum, there are different layers are in present in the rocky substratum. One is older layers, another one is called it as younger layers. Normally the species are dead species normally present in this area. Okay. Then how the decide the age of the fossils? So there are different methods are used. You see radiometric, luminescence, dendrochronology, amino acid dating. So radiometric means by means of isodepes, then luminescence by means of heat or lights, then dendrochronology. You see the diagrammatic representation for the tree. So inner rings, there are different rings are present. One ring developed for the each year developed one ring. So according to this, okay, you see how many years this tree, okay, we see that. Then another one example, this is the fossil for the Archaeopteryx. This is a transitional species because this is example for the connecting link. Archaeopteryx is the connecting link between um, reptiles and birds, because Archaeopteryx which contains interconnections. Okay. Then next see the homologous structures. Homologous means what? Anatomically different. The structural aspects is different, but they have same functional aspects. So the functional aspects remain same, but the structural aspects will be differ. That is called a homologous structure. Okay, this is one example for what? Evidences for the evolutions. Then analogous structure. Okay, this is analogous structure. What is analogous structure? Analogous structure is different, yes, but related functions. Okay, the different closely related functions, that is. Then see the vestigial structures. Vestigial structures is nothing but what? Some vestigial organs are present in our body. So many vestigial organs. One is example for the for what? Appendix. Appendix is an example for the vestigial organs. So normally this is well developed in ruminants. Actually, ruminants is nothing but cow coat, etc. So herbivorous animals, appendix is well developed. In herbivorous animals, appendix is well developed. This is one example for the evidences for evolutions. Okay. So next we come to the embryological evidences. So, so you see the diagrammatic represents there are different embryos start from fetus to human beings. There are different embryos, but they have some similarities. Structural similarities are present. Okay, you see some structural similarities is present. So normally, one example how the evolutions travel. We can provide some example. We can provide some evidences. Simply take it as heart, the development of heart in the embryo. So normally, fishes have a two-chamber heart. Fishes, amphibians have a three-chamber heart. Reptiles, three-chamber heart, partially divided ventricle. Then in aves and mammals, a four-chambered heart is present. So the evolutions from fish, amphibians, reptiles, aves and mammals. So this is the embryological evidences. You see, all the embryos are remain same in top of the portions, but in adult each and every will be differ. Okay, but this condition is called biogenetic conditions or biogenetic law. Ernest von Haeckel produced the biogenetic law. He told ontogeny recapitulates biologeny. Ontogeny is nothing but the development. Phylogeny is nothing but their ancestors are their okay, their ancestors alone. Okay, so one organisms which crosses the the developmental stages crosses its ancestors, that is called ontogeny recapitulates phylogeny. Say for example, the presence of gill slits. In presence of gill slits in all species gills are present, but we are not having gills. The gills only present where? So this is in fishes only having the gills. That is this is called the embryological evidences. Okay, so yes, so the embryological evidences which, yes. So next molecular evidences. So we can read the DNA and amino acids from the ancestries, evolutionary ancestries. So normally amino acids difference from human beings hemoglobin. Okay, in gorilla 1, monkey 8. So number of amino acids is different actually. So more than 200 amino acids are present, but only the difference, what is the difference? You see, so the evolution is fish, then frog, amphibians, then what? Aves, then actually mammals. Okay, so this is the ancestry. So we can read the DNA, amino acids, etc. Okay, so these are the example for the evolutionary evidences. So let us see some photographic evidences. So fossil evidences. This is the mammoth elephant in Siberia. This is actual remains present in the Siberia. So Siberian elephant, it looks like this. Okay, Siberia is the colder regions. So the actual remain is present for a long time. So this is one example for the fossil evidences. Then this is the actual remains. So normally petrification is present. Petrification is nothing but and the sedimentation of the rocks. There are different layers already we had seen in the video. Okay, so this is a petrification is nothing but the remains. Okay, so next the organ, homologous organ. What is the homologous organs? What is? Okay, good. Homologous organs is nothing but the structural similarity, then they are having the different functional aspects. The structural similarities is you see the metacarpals here is the also presence, so that is a structural similarity. Yes, in part, what is the purpose? Yes. Yes. So what is the purpose? Flying. In porpoise is nothing but whales. Whales for swimming, horse for running, walking. Okay, frog hopping. Okay, so that is the example for the homologous organs. Okay, so the structural similarity, functional dissimilarity. Structural similarity, functional dissimilarity. So likewise in plants, Fogainilla, Cucurbita. So what is thorn and tendril also homologous organs, because the structure is same, remains same, but the functional dissimilarity. This is productions. This is for the supporting. Okay. So next analogous organs. We see the analogous organs. You see the fins, wings, flipper. Okay. So here analogous organs, all the structures are responsible for swimming, but the structural differences are there. Okay, so analogous is nothing but structural dissimilarity, functional similarity. So likewise in plants you see. Okay, so this is the example for the analogous organs in plants also. Okay, right. So next this is peripatus. Peripatus, this is Archaeopteryx bird. Peripatus is the connecting link between Annelida and Arthropoda. So annelid, what is the characteristic features of annelids? Annelids, they are having worm-like structure. They are having antenna. So antenna is the characteristic features of arthropodans. This is one example for connecting link. So by using the evidences of connecting link, we said from the annelids arthropods are developed. Okay, right. Archaeopteryx already I denoted. Archaeopteryx is the connecting link between yes, reptiles and birds. So from which we denoted, so birds are developed from the reptiles. This is one example for connecting link. Then another one is atavistic organs. Atavistic organ is nothing but the vestigial organ sometimes developed in the organisms, that is called atavistic organs. So here is one example, the development of the human embryo, human fetus. Here the tail is developed. Here the tail is developed. So the tail is the atavistic organ in human embryo. Right? Atavistic is nothing but the vestigial organ going to be developed in the fetus or embryo or the child, that is called atavistic organs. Okay students, so today class we are discussing so two concepts, One is called what is called geological time scale. Another one is called what is called yes, the various types of evidences supporting to the evolutions. What are the evidences? Anatomical evidences, then paleontological evidences, then molecular evidences, yes, embryological evidences. Okay, students, study well. Let us see in next class.